At this time, I'd like to introduce today's host, Guillermo Ceniza Bordallo. Guillermo is a predoctoral researcher at Complutense University in Madrid, Spain, having previously achieved his bachelor's degree in physiotherapy and a master's degree in health sciences. His research focuses on unraveling the mechanisms underlying persistent pain in the pediatric population following surgical procedures, specifically chronic post-surgical pain. With that, I'd like to pass today's proceedings over to Guillermo, who's going to introduce us to Gabriel, and I look forward to hearing from everyone. Thank you so much, Guillermo.
Okay, thank you so much. You're very welcome to be here in this webinar. It's a pleasure to me to present Gabriel Reiner. Gabriel is a PhD student in data science in University of California, San Diego, US, and achieved a bachelor degree in neuroscience from the University of Southern California in US too. He has contributed to research in pain neuromyelin and now focuses on brain imaging statistics, including developing new analysis methods and open source software. So it will be a pleasure to be here with Gabriel and to learn more about his works. Thank you very much to be here, Gabriel.
Thank you, Guillermo, and everyone in attendance. Looking forward to this webinar. Let me share my screen. You can see the slides?
Yep, looks great, Gabriel. Thanks.
Okay, great. Okay, so in this pathway to publication presentation, I'll be presenting on a paper I published about two years ago in pain, and it involves the neuromodulatory systems that are involved during mindfulness-based pain relief, and specifically linking mindfulness meditation pain relief to functional connections involving the sensory thalamus. And then I'll end with how this research project impacted my academic interests and my studies now. Okay, so just a quick overview of the study. There were 40 healthy subjects that underwent pain testing using a thermode to deliver a series of on-off plateaus of 49 degrees Celsius to the back of the calf. Pain intensity and unpleasantness ratings were then collected using an 11-point visual analog scale. And in conjunction, fMRI data were acquired. Okay. So during the fMRI scanning sessions, firstly, there's a baseline testing session. Then the groups were randomized into 4-session interventions, either a mindfulness training session or a book listening control group. And then the scanning session occurred. So during fMRI scanning, scans were collected during rest and heat stimulation. Then these scans were repeated, and the mindfulness meditation group meditated while the control group continued to rest. So this is during heat stimulation. Okay, so just a quick overview of the behavioral results. So this is during the fMRI scanning session. There's an interaction on the visual analog scale pain ratings that was driven by this 32% reduction in pain intensity during meditation when compared to the rest and control groups, as well as similar pattern for pain unpleasantness. Okay, so taking those changes in pain scores and correlating them with changes in brain activation, regression analyses were used to determine where in the brain mindfulness-related changes in pain correspond with changes in brain activation. And the greater the reductions in pain intensity, the greater the deactivation in these sensory areas known to process pain inputs. So this is not too small to see, but see deactivation of posterior insula, anterior insula, ventromedial prefrontal cortex. And these are associated with reductions in pain intensity. Okay. And then the brain connectivity results. Pain relief here was associated with decoupling between the thalamus and the precuneus, which is involved in integrating sensory stimuli with subjective experience. And these findings are the first to show this endogenous thalamus precuneus pain modulatory mechanism. Okay, so this publication, was about two years ago, and since then, this was before I was in grad school, and then I started a PhD program in data science. And my interest in pain neuroimaging were originally driven by its compelling philosophy and its clinical relevance, and understanding how the brain processes pain, and how techniques like mindfulness can relieve pain. It's not only something that's mechanistically interesting, but also has potential to help patients suffering from chronic pain. And the philosophical and clinical intersections of this field drew me in during my time working in clinical trials and data analysis. And specifically this paper, which I just presented, really sparked my interest in methods and neuroimaging analysis methods more specifically. So I became interested in how we can extract this meaningful information from fMRI images and process complex signals from the brain. Also, this experience with small study neuroimaging also showed me the limitations of what you can and what you can't learn from small scale neuroimaging studies. So there are, of course, reproducibility challenges in all areas, but notably neuroimaging studies. One of these biggest challenges is that for statistical summaries of small fMRI datasets often depend on arbitrary model selection and signal processing decisions that you make throughout the signal processing pipeline. And these small decisions can significantly affect the final results or the final images that you would report on in a publication like this. So this can make it difficult to trust findings, and it led me to rethink where I wanted to focus my research. So this shift in focus, I wanted to improve more the reliability of brain imaging research. So I need to dive deeper into the statistical models and the software that powers these neuroimaging applications. I transitioned into a data science PhD program. And now my goal is more to build on the foundations of data science to develop better methods and tools for neuroimaging, and also strengthen my understanding of the math and theory behind how these models are fit. So I think the role of data science in pain research is someone who can bridge the gap between domain knowledge in pain and the increasingly complex data sets we work with. So pain research can involve a lot of outcomes, as we all know, from patient-reported outcomes to neuroimaging data and omics, qualitative sensory testing, etc. So data science can provide the tools to navigate this complexity and enables us to extract meaningful insights from these diverse data sources. So for new methods to be accessible to the broader research community, I find that writing software and contributing to widely used open source packages can have a greater impact, at least in my opinion, than publishing a paper, because you can get many researchers might be interested in using a software, rather maybe a few would read a paper. So now I helped develop or contribute to open source software that exists, and NiLearn is a Python package for machine learning from brain images. Aeon is a similar package, but it's for time series more generally, so it applies to multiple domains. And then NeuroMaps is an interesting package that compares using different methods to compare spatial brain maps. So just to conclude, the process of writing my first paper did help me realize both the limitations and the opportunities in pain neuroimaging. And it kind of pushed me to transition into data science, where I'm now focused on developing the tools and methods that can be used to improve reproducibility and reliability of brain imaging. And open source is important for this advancement, and I'm excited to be part of that community working towards that goal.
Well, thank you very much, Gabriel. Well, we are super excited with this first work, and feel free to ask question in the open question button that you have in the screen. Thank you so much for this presentation. It will be awesome. And in the end of the third panelist, we have time to ask questions and to discuss more about that. So thank you so much, Gabriel, for this incredible work. So, okay, now we have the second panelist is the Dr. Alison Barry. Alison has a research background in sensory physiology and bioinformatics, having achieved her PhD in philosophy at University of Oxford in UK with David Bennett and Georgios Vasco, looking at molecular profiling of murine primary afferents in neuropathic pain. Also, Alison currently split her time with the University of Vienna in Austria and University of Texas at Dallas in US, using multi-omic methods to understand pain pathophysiology. So thank you so much, Dr. Alison, to be here today, and we are super excited to hear you.
Great, thanks so much for having me. So I'm Allie. I'm a postdoc, splitting my time currently between the University of Vienna and the University of Texas, Dallas. A part of that reason, kind of was briefly kind of introduced, is that my main kind of interest is on multi-omic data integration, and so I figured the best way to do multi-omic data integration is kind of to sit at that intersection of everyone that's generating different types of data to get to be the person to then play with all of it and bring it together. Today, I'm going to talk about a paper that came out of my PhD work at the University of Oxford that was looking at deep RNA-seq of murine subtypes, sensory neuron subtypes after nerve injury. And so kind of very briefly as an overview of the project. So our main research question here was we were hoping to explore the transcriptional signature or the RNA-seq signature within each of the different sensory neuron populations in the context of sex and injury. And so there's a couple of reasons for this, but I think the big thing that kind of really interested us was this idea that we have such diversity in the primary afferent systems, and we don't have a great understanding of how each of them respond differently in the context of pain. We've had reports come out that, for example, you have low threshold mechanoreceptors, which are traditionally involved in touch, which after injury can contribute to mechanical hypersensitivity. But then the underlying molecular profiles of that aren't clear, and are they changing in the same way that maybe your nociceptive populations are changing, and are the different nociceptive populations kind of changing in the same way at a molecular level? And so this project really set out to kind of address that question. On the technical side, when we think of RNA-seq or transcriptomic studies, there's kind of two main approaches. So you have kind of single cell or single nuclear or spatial RNA-seq, and then you have bulk analysis. And so when you're looking at kind of single cell analyses, they really, really benefit because you can look at all the different cell types that are in your tissue. And so that's a huge strength. One of the downsides, however, is that there's so little material per single cell to sequence that just technically we're not really at a point where you get exceptional coverage of every gene in every cell. And so what ends up happening when you're doing your downstream analyses is you typically will cluster all your cells together that look like they belong to the same cell type, and then you kind of treat them almost as a pseudo bulk. We've kind of merged these groups together, and then you can do downstream analysis. And in the context of injury, we know from previous reports that some of the cell types actually seem to lose almost their cell type markers. So there's this concern that actually after injury, you're not going to be able to cluster things in quite the same way, to quite the same efficiency. On the other side, we have bulk RNA-seq, which is great in terms of kind of the number of genes that you're detecting, and especially in the context of lowly expressed genes. But obviously you're losing any heterogeneity in your tissue because you are just kind of blending everything together. And so this project really set out to search for that middle ground in the context of primary afferents and see if we can find out how to do almost bulk quality RNA-seq, but on subtype specific samples. And so the overview for this project, we relied heavily on mouse transgenics to do this. And so the benefit with mouse transgenics in the field, fortunately, is we have decades of individuals here who have been developing markers for the different subtypes that we're interested in mice. So we can actually take a driver line, cross it to a fluorescent reporter line. And what you end up having is you have kind of in each of the cell types of interest, depending on what transgenic you're using, you actually have fluorescent reporters exclusively in that cell type that you're looking for. And so we use this across 5 different cell types of interest. You can then separate out the cells that are of interest based on their fluorophore expression and almost develop kind of a bulk sample based on this sorting mechanism. And so we were able to do this. We did this for kind of a general nociceptive population or a nociceptor-like population that can be labeled by NAV 1.8. We did this for peptidergic nociceptors. We did this for non-peptidergic nociceptors. And then we also did this for 2 of the low threshold mechanoreceptor populations. We looked at CLTMRs, which in mice can be labeled by TH, although that's not a great marker for them in humans. And then we also looked at A beta and A rapidly adapting LTMRs, which can be labeled by TRAC-B or NTRAC2. And so the study design here, we did a nerve injury model called an SNI or a spared nerve injury, where you transect 2 of the branches of the sciatic nerve. And then we waited either 3 days or 4 weeks across males and females and isolated the tissue at those time points to compare both the injured side to the uninjured contralateral side. In terms of kind of general research takeaways, kind of in the biology side of things, so we were able to pull out some sexual dimorphism in the contralaterals, almost naive-like subpopulations before we considered the injury at all. And so this was showing that predominantly while you're maybe expressing like the sex-linked genes to be differentially expressed, we do pull out, especially in the TRAC-B population of low threshold mechanoreceptors, a bit more sexual dimorphism in that population that could be functionally relevant. We then went on to look at shared signatures of injury as well as subtype-specific injury signatures to see that maybe the TRAC-B population by 4 weeks is actually doing something different. And then there's also some differences maybe in our non-peptidergics and CLTMRs where they're behaving uniquely from our peptidergic populations at a molecular level. And so this then kind of gave an avenue for follow-up studies as well. We also went on to look at this idea of interaction between sex and injury to see, because we have so many sexually dimorphic conditions, especially clinically, we wanted to see that at the neuronal level specifically, preclinically, are we able to pull out those changes? And it doesn't seem that at least interaction-wise, there's many large differences, but this likely just highlights that it's probably non-neuronal cells that are frequently driving this, which is, I think, in line with a lot of the previous literature looking at inflammation and kind of non-neuronal glial cells as well to show that they're very, very important in terms of the sexual dimorphism that we're seeing. I think another really big takeaway that came out of a project like this is that you generate a lot of data when you sequence 160 samples, and it also takes a really long time to collect. And so doing something with the data that's more than just putting it in supplemental tables in a paper became very, very important to us who are working on the project. And so this is a lot of work that a wonderful undergraduate student at Oxford, now a master's student, Lin Zhao, has worked with me on, in that we didn't want to just look at the data sets in the context of kind of the 6 main figures in the paper, but we wanted to really expand this into a resource for the field. Because at the end of the day, I think the data accessibility and availability to everyone is important. We spent a lot of time collecting the data. We want people to be able to use it effectively and efficiently. And so having a website where people can very easily kind of query their genes of interest and see how they change, I think is a very kind of easy way to go about doing this. So we've kind of joined groups of individuals in the field who are starting to build databases like this. I think one thing that's almost kind of evolved from then is we've developed this database quite a bit. So it started in how we published it with this paper was just on our single data set. It's now hosting over a dozen data sets from the neural injury group in Oxford. And so this is again work that was really championed by Lynn in the group. And so we've now got a mix of species on the website. You can query kind of genes in the context of all of the data sets, or you can start digging into some of the data sets more nuanced to see kind of within each of the different data sets. For example, we have, you know, human skin from a specific cohort. We have induced pluripotent stem cells from a different study, as well as some mouse and rat DRGs. But really kind of focused on this idea of the data exists and we want people to be able to use it as much as possible. And so there's got to be a better way than what we're doing just in the context of a single paper, but really kind of making sure that these resources exist. And we've expanded this since. We've written a couple of grants to hopefully expand kind of outside the scope of just the lab. I've also been working for a bit with the NC3Rs, looking at this idea that actually, if you're going to use animals in your research, almost a responsibility of that is to make sure that the data is used effectively. And so something like open data sharing can really help kind of meet those targets of effective use of animals. We've also been able to spin out the paper into a couple other studies. And so a complementary study that was going on kind of alongside this initial paper that came out in Pain recently. And this was this kind of idea that the different cell types actually do respond differently to injury. And so this was work that was done in collaboration with Greg Weir's group up in Glasgow and was led by Andy Cooper, who did some exceptional whole DRG counting to show that the non-peptidogics and the CLTMRs do in fact actually respond differently than some of the other cell types. And then on the data sharing side, we've expanded this further. And I think this is something that I think links quite well to what Gabrielle was talking about earlier. And it's this idea of kind of your code base availability. And so we've taken the database slightly further and we've put together almost a user manual, both for people in-house who are kind of maintaining the database at the moment. Even though I don't actually work at Oxford anymore, I do still maintain our database that's hosting all of the data, and we'll continue to update it. But we also wanted to provide a guide for other people who are maybe looking to use this code base, develop resources like this themselves, and make sure that it is properly documented. Really in this push for kind of open collaborative science. And so with that, I'd like to thank everyone involved. So especially at Oxford, who was involved directly in the study. So Dave Bennett, Georges Viscozas, as well as Lin Zhao and Jun Yang, but also my two current institutes at University of Texas, Dallas, as well as University of Vienna.
Thank you very much, Dr. Barry. It will be incredible work, and we're super happy to be your work here. Thank you very much. Only one thing important, you have box in the bottom to ask questions to our panelists, and feel free you have questions or yes or whatever. After the presentation, we have time to ask questions, and feel free to use it. Thank you so much, Alison. The third panelist that we have, it's a pleasure to present Dr. Hannah Jennings. Hannah is a clinical and health psychologist working in the eating disorder program at the Hospital of Sick Children in Toronto, in Canada. She received her Master of Art and PhD in Clinical Development Psychology at York University in Toronto, also in Canada, where she completed her research in opportunity to understand childhood heart laboratory under Rebecca Pillai Rhythm supervision. Hannah also researched focused on early childhood pain-related distress, notably how caregivers support infants to regulate their pain during routine vaccination, as well as how children's own regular strategy during vaccinations develop to motherhood. Her graduate research has been published in several high-impact journals, including her dissertation works being selected as editor choice in Pain. So it will be a pleasure to have here Hannah today. Thank you so much, Hannah.
Thank you for having me. Sorry for the rapid spinning through the slides there. Still have tech challenges after doing the virtual world for so many years. So I'm very excited to be here. Thank you for having me. It's been a pleasure to sit through the other presentations. I am going to talk a little bit about my pathway to publication and impact, and really focus on tips that I have for how to do so based on the journey I took to get to this paper. So the paper I'm going to be speaking about that was published in Pain is part of my dissertation work. And the title, as you can see, is called Understanding the Concurrent and Predictive Relations Between Child-Led Emotion Regulation Behaviors and Pain During Vaccination. So in toddlerhood. So rather than kind of going through the entire methodology, I really just kind of want to ground this in these tips and kind of walk you through how I followed those tips and how it got me through my methodology to ultimately get to where I landed with the paper. So the first tip I have for you as early career researchers is to let passion lead you. Chances are, if you are doing a doctorate, you have found a field that you're quite passionate about and want to spend quite a bit of time studying. But my journey was a little bit different in that pain research had always been my passion. As a researcher, I had started in my undergraduate degree at Dalhousie University studying pediatric pain. And this continued on when I went to York University to get my master's and PhD in clinical psychology. And so pain had been a long-standing passion of mine, but along the way, because I was in both a clinical and developmental psychology program at York, I also spent a lot of time diving in and digging into the way emotions developed and emotion regulation develops in early childhood. I studied early childhood and vaccination. That was the bulk of the work we did in the Ouch Lab. But I also became quite interested in using that context to better understand how children's self-regulation skills develop. So when I immersed myself in both of those literatures, the child emotion regulation literature and the pediatric pain literature, I noticed two key gaps that could actually complement one another. And so what I was noticing in the child emotion regulation literature when I took that leap into that literature was that because they were all based on experimental paradigms, we actually couldn't ethically study very high levels of distress, because those of you who have done research with children, you know that ethically, we tend to stop procedures when the distress becomes unnecessarily high. And so we actually could really only study low to moderate distress. As a pain researcher, watching hundreds and hundreds of videos of kids getting vaccinations, we know that distress can go much higher than moderate. And so we felt that our vaccination paradigm could complement what was being done in the developmental psychology literature. But at the same time, the pain literature was really missing the opportunity to look at, in kind of the first couple of years of life, how children's own emotion regulation skills developed and what kind of behaviors they were doing to calm themselves down during vaccination. A lot of that work was happening in older populations and kind of school age kids, but we know that this develops in kind of toddlerhood, and that was the population we were studying. So I actually developed a new passion, which was to kind of marry or merge the two fields, pain and developmental psychology, to see if the other field could complement the gap of the other. And that is where kind of a new passion evolved for me that got me really excited to do this work. This brings me to my next tip, which is really to take your time and dive in. So prior to getting to this paper, which was the second paper of my PhD, because we didn't really have a starting point in the pain literature, we had to do a very deep dive into the emotion regulation child development literature, which was the first paper of my dissertation, which was a systematic review of the first 3 years of life and the relationships between what children were doing to regulate their behaviors, so those emotion regulation behaviors, and how it related to their distress and these experimental paradigms. And really the hope of that was to see across kind of the first year of life, second year of life, which really marks the start of toddlerhood, and the third year of life, were there any differences in what was helpful when kids were distressed? And what that allowed us to do, I see a typo on my slide, I'm sorry for that. But what that allowed us to do was also look at what measures are people using? Like, do we even have a starting point here to bring this into the pain context? Because we didn't really know how to begin. We didn't know who to reach out to, who to collaborate with. We had many pain researchers we could collaborate with. But again, we really needed somebody who was an expert in developmental psychology and emotion regulation, to be able to take that step forward and bring something new into our field. Which comes to tip number 3, which is building relationships. And I don't necessarily mean collaborating within your field, within the area of pain, but not being afraid to reach out and build those relationships with experts in other fields who can teach us something in pain. And so in that systematic review, we found that the lab tab measure, which is a temperament and emotion regulation battery of experimental paradigms and coding systems, this tended to be the one that was most commonly used or incorporated behaviors that were used by other researchers in a much more formulaic way. Goldsmith and Rothbart are the kind of originating creators of that measure. But it was now being housed at Texas A&M University. And Dr. Jeffrey Gagne was the individual who was responsible for training and making sure that the coding systems and the paradigms were being done properly. So we reached out. I reached out to Texas A&M, to his lab, and I said, you know, we're very curious about this measure. We really want to bring it into the pain context. Where do we even begin? How do I even get my hands on this manual? Like, I need to read the thing. I've read about it in papers. How do we do this? And it really was one email that started this process of really working with somebody who was an expert in this other area and combining our expertise in pain and child emotion regulation to come up with a measure that we could bring into our lab, to bring into the vaccination setting that would allow us to better understand how children's emerging emotion regulation behaviors could impact their pain response during routine vaccinations. I think what I want to highlight is allowing yourself to do something different and difficult. And I say this with the caveat that I had the privilege of working in a lab where there was already a lot of data collected, already a methodological process underway that allowed me to really pick this one particular battle to focus on and put all my energy into. And so the battle I chose was to bring this measure into the lab and really focus on filling that particular gap. There are many battles you're going to come across, whether it's in your dissertation, whether it's in other parts of your research career, and you're not going to be able to target each one. And I want to acknowledge my privilege in the fact that I was able to feel comfortable that I had a process in place and a supportive lab for being able to do just that and to focus on this one area. And I say that because this took a lot of time and it wasn't the easiest path forward. You know, doing a dissertation or postdoctoral research, you are creating new knowledge. That is the standing, that is the basis of moving forward into a research career. And I'm sure there was an easier path. And because I followed tip 1 so closely and was insisted on following that particular passion, and I had the ability to do so, I really took my time with it. I really took my time to get into the weeds with our new collaborators and really think through how we can bring this lab tab variation into the vaccination setting. I created a team of coders. They were undergraduate students who are wonderful, and they were with us throughout the entire iterative process of going to the drawing board, looking at the measure, thinking about what worked and what didn't work in the vaccination context, working with my supervisor, Dr. Rebecca Play-Riddell and Dr. Gagne, and looking at our video footage of vaccinations and talking about why certain things worked and certain things didn't. And we worked towards reliability, and that took a lot of steps forward that unfortunately resulted in a few steps back. So that classic 2 steps forward, 1 step back, that was a very significant part of my journey, because we thought we had something, and then when we put it into practice and tried to get reliable, because we were modifying this, because we were developing this, we had to take some steps back and recalibrate. And that all took time and support, I should add, of a supportive supervisor and lab. And so it was a process, and it was well worth it, because in following these tips, it allowed me to kind of get to this point and bring forward a manuscript that was both impactful for the pain research world, because it was one of the first times, if not the first time, where we kind of formulaically looked at the measures in another context and brought it in to inform what was going on in toddlerhood and how we could study emerging child regulation during vaccination. And it also provided a context for developmental psychologists to experiment with high distress settings without the fear that they had to stop procedures, because as much as we try, we ultimately can't change what's happening once the needle goes in. We can support parents and children to use their skills, but ultimately the needle goes in it, and that is that. And many of us have been working towards figuring out how to make that hurt less, and we've come a long way with that. But briefly, in terms of our methodology, I've referred to videos. So we would go into doctors' offices in Toronto, Ontario, and we would video record parents and their toddlers getting their vaccinations at what we call well child visits. And we would bring those videos back to the lab, and we had my team of coders coding the lab tab measure. And we looked at particular clusters that were identified in the systematic review I talked about. So their use of disengaging their attention, use of physical self-soothing, and use of parent-focused behaviors, which was actually them reaching out to their parents for additional support. We hypothesized because they've learned in the development of the attachment relationship that when they're highly distressed, they can turn to their parents when they're having a hard time calming themselves down. And so we expected that we would see some of this in this context. We also had another team coding pain-related distress using FLAC. That was a separate coding team. And we looked at the concurrent and predictive relationships between the two measures. So concurrent being at the same time point within the needle appointment, and predicting forward. So we looked at 1 minute post-needle, 2 minutes post-needle, and 3 minutes post-needle. So the concurrent ones would be within the minute, predictive would be predicting forward. And we looked separately at 12 and 18 months because, again, our systematic review showed us that there were some slight developmental differences across points in early childhood. So what did I find? So looking at our 12-month-olds, we found, consistent with what we learned in our systematic review, looking at the broader developmental psychology literature, we learned that physical self-soothing was helpful at reducing pain-related distress. So we saw that when kids were using more physical self-soothing, their pain was lower concurrently. At 18 months, we did see the same thing about physical self-soothing, but actually it was to a lesser extent than at 12 months. And we weren't surprised by this because we would expect that at 18 months, as they emerge into toddlerhood, their self-regulatory abilities, their self-regulation behaviors might become increasingly complex, whereas earlier in toddlerhood, just at the cap of infancy at 12 months, they still might be relying on more primitive physical methods of self-soothing during vaccination. And at 18 months, we actually saw that they were disengaging their attention more, and this was helpful at reducing their pain-related distress. And they were signaling to their parents when they were experiencing more distress, hey, I need your help here. Like, I'm doing what I can, but I still need you to step in. And that sort of dyadic dance between parent and child that we learned about at 18 months with this parent-focused behavior actually informed how we moved forward with the final paper of my dissertation. And just of note, the majority of the relationships were concurrent, so we didn't see as many relationships predicting forward. But in our third paper, we looked a little bit more closely at longitudinally over time, the slope of pain-related distress across the needle and how parent behaviors and these emotion regulation behaviors impacted that decline. So that is another paper for another day. So all that to say, this was a huge body of work that I'm quite proud of, but I really did kind of follow these core tips that I would recommend for any project, for any path that you move forward on, to let passion lead you, to take your time and dive in, to build those relationships, and to not be afraid of doing it outside of your field, and to allow yourself to do something different. And that's all I've got for you today.
Thank you very much. It was super, super great to hear you, and these tips are super, super useful. Yeah, thank you so much. We have now a moment to your questions. You have a box down. So I will start the first one that I have. That's personally my question, but I think it's a point that maybe everybody are like in their minds. So the question is, what's the biggest challenge that you find during the publication process in pain, and how did you overcome it? So okay, if you wanted to start, Hannah, or
Sure.
yeah.
What was the biggest challenge? Oh, if I'm speaking frankly, to have the confidence to do it and to put in the work. But truthfully, I think the most challenging part of it was not taking shortcuts, like really thinking things through at every step of the way, you know, really like we're so busy in all of our careers and our fields and our studies. It can be kind of really hard to balance it all. And I think it can be hard when we're so excited about things and we want to take on so much. I think the biggest challenge was on the one hand, I want to say not taking the shortcuts and thinking it through, but also on the other side, I think a challenge is not making it so big. So like on the flip side, when you're so passionate about something, you kind of want to bring everything in. And my supervisor had to kind of pull my reins back a little and say, whoa, 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 like you've built something, slow down, like think it through. And then on the flip side, also not, getting exhausted and just wanting to reach the finish line. So I think on the one hand, it's that tug and pull of, just wanting to do it all. And then you do hit a little bit of a point of where you're exhausted, especially in my context, as this was, a dissertation and what felt at that point like a lifelong work. Just really studying yourself and thinking things through and doing it justice when you get to the time where you're submitting it and thinking it through. So off the top of my head, that's the challenge that's jumping out for me, is that kind of balance between the two.
Thank you. What's your opinion, Alison?
Yeah, I mean, to be honest, I think I'd agree full-heartedly with what Hannah said. I think it's really hard in projects that at the end of the day, you do want to kind of see it through fully. I remember even in the context of our database, it was one of these things of like, I mean, my sample collection also, for context, like it had been planned in the spring of 2020, which as we all know is like a superb time to be doing wet lab work, just when everyone wants to have things planned. And so I ended up largely having to redo the entire data collection for the project. And so by the time it got to the end, I was like, I will make the most of this data. And you do have to scale back a little bit. It was even this consideration of like, do I have time to like build a database to host the web? And I think for me, it became that challenge of like, what is my priority here? And like, where do you balance those? And so I made the choice to invest the time into building a website to host my data because that felt like it was worth it in that case. But then there were other things for the project that just didn't seem to weigh the same. And so it really was finding that balance of you're never going to be able to do absolutely everything. And so where do you draw that line for yourself and for the field, and what's going to be the most useful for people that you're working with?
I think it goes back to picking those battles, right? And thinking through like, what is the battle I'm going to like fight on and stay.
Yeah, absolutely.
Okay, Gabriel, do you have any comments, things to add?
Yeah, I definitely agree with the comments on kind of choosing choosing a path or a set of methods that isn't too big, because publication does take a very long time. In my experience, it's often the case, like you have a research question, you collect the data, you do the analysis, and then essentially you have the results, but that's only part of the process. Then the process of writing up can be super lengthy, and it's oftentimes difficult, or in my experience, it's difficult to stay motivated through multiple iterations of a draft and then get comments back. And by that point, you kind of, in your mind, moved on to other projects and new things. So just, I think, being prepared that things take a long time, even for small projects or for papers that seem straightforward on the onset, everything takes a while. So that goes in line with not taking on too much. And even if you have just like a result that you don't think is enough for a paper, it oftentimes is enough if you just look at that result in detail and analyze it fully. It takes time, and the scope is important.
Yeah, absolutely. This is a challenge. I think this is the most important point. Yeah, absolutely right. Okay, we have a comment and question now from Don Daniel. He says that it's a great presentation for everyone. He has a questions. He said, we know that pain is a high impact pain journal. And the question is, do you take actions post publications to ensure further or increase the impact change in knowledge users?
That's a really good question. I'm trying to think back to when the paper got published. We shared it widely. So, our lab, Payne, my supervisor, myself, we were sharing it quite broadly. We were kind of giving snippets, on Twitter, for example, to share and get it out there. And we did try to promote it as much as possible, which I think was hard for me. I'm not necessarily the best at promoting. So that was a bit of a learning curve for me, just in terms of, being asked to do the video for Payne to attach to the editor's choice paper and all these pieces. It was well worth it, and kind of intimidating, to be honest, to put yourself out there, to put your face literally on the paper at a video that people could download and have. I'm not going to lie, actually, I could probably say that was one of the more challenging parts for me. And it is exciting. Like it's a practice in allowing yourself to be very proud of what you did. And I find as researchers and clinicians, for myself, many of us are humble or like to try to be humble, and it can be kind of hard to put ourselves out there. And it was an important way to get the paper out there and get yourself out there as a way to network and sort of collaborate and build more relationships with others.
Yeah, I guess
Yeah.
just kind of jumping in there as well. I think one of the things from our side, so we also took the, Twitter promotion route, which I think, and I mean, maybe now if we're kind of all shifting towards blue sky, but I do think that there's something in it to kind of be able to have those discourses in a really casual space. The other thing that we've started doing and is something that we definitely need to improve in is that I'm very fortunate that during my time at Oxford, I was a part of a very large consortia that worked very closely with patient partners. And so was able to present the database to them as well and get feedback there. And now it's a little bit of like, well, the database realistically is not actually accessible to patients, even though it's something that I think a lot of people have expressed that they would be interested in kind of understanding how it works and what the data underneath is. So now it's kind of taking this back to the drawing board and be like, okay, how can we then do better in the next steps and make sure that this actually does involve the people that fundamentally this research is for at the end of the day.
Mm-hmm. That's a really good point, the importance of knowledge translation and getting it out to people in a way that is accessible.
Mhm. Yeah, I agree with the points here. And what the Payne paper that was published, I was in a lab that was very good at this outreach, because personally I don't feel like it's one of my strengths, but there were some like news articles to the university that helped kind of advertise the paper and create discussion on it. And then one other thing I think is important going forward, similar to Allison's approach at creating databases from the study, is that the paper is more of like a static object that people will read, and then they'll either remember it or cite it in their work. But also if you have like something more tangible, like this database approach, or if you create a new analysis approach, if you have the code to recreate that, that can have also lasting impact for other researchers that want to use similar methods in their work.
I wanted to offer just a quick contradictory view, which is a little weird given my current position, but really self-promoting and getting my work out after grad school. I was fortunate enough to be able to publish in scientific reports in the prostate, but this was something that my lab at the time when I graduated was not very invested in, and we didn't, in my opinion, do a tremendous job of promoting the work. And to Hannah's point, I was more than happy at the time to take the humble approach, but I've certainly switched course as I've grown older, and there are so many avenues to explore to promote yourself and to connect with like-minded folks. So please, if you ever have any questions, there's a role of IASP here, and you can certainly reach out, and we'd be happy to collaborate with you. Sorry for poking my head, and I appreciate it, Guillermo.
You're very welcome for sure. Thank you very much. I think maybe we have time for one question more.
Sure.
Yeah, I think okay. The question that's for me. I think it's pretty important to know if yeah, if your publication, the article that you publish on Pain, changed in any way your opportunities or your career, your trajectory, and if this article opened a new opportunities for you.
That's a good question. I think currently I sit in a clinical position, and I often think back to this paper and the impact it had on me as a researcher. And I'm actually currently in the process of re-engaging in my research skill set. And I think, I don't know how to exactly word it, but of course, publishing in high impact journals helps when it comes to, applying for grants and continuing on that trajectory. But when I've taken this pause, that paper has actually helped me kind of go back and sort of build my confidence to get back in there again, because I took more of that clinical path when I have an equally strong research path. And so personally, I've kind of gone back to it, and I'm grateful for this opportunity because it also kind of forced me to get back into this paper. You know, it reminded me of that kind of research passion that I have, which could very well soon turn my career trajectory on its head a little bit. We'll see where that goes, but ultimately it will help in terms of having these types of high impact publications, whether it's an academic career, whether it's trying to have, if you're a clinician, sort of a 50-50 or kind of some other split type of position. I think having these types of publications have meaning, you know, to our research colleagues, and I think it'll help moving forward.
How about you, Alison?
Yeah, I think, well, for me, this project especially, I think at the time, so I'd worked with omics data before, but this was by far the largest data set I had worked with. And I think just in wanting to do it justice, almost set me on this path of multi-omic integration, and this idea of like, we have so much data. We've gone through almost a decade of this expectation, and on the preclinical side, that you're going to add in an RNA-seq data set to whatever study you have. And maybe people are adding like a single volcano plot and like moving on with their lives. And it just, there's so much in each of these data sets. And so it's kind of forced me to deep dive, and I think in a good way, into the like, okay, I mean, I am still generating data, so this isn't exclusively that, but it's the like, of the data sets that are already out there, like how can we maximize their use, not just in the ones that I'm working with, but across the board. And so that's, I mean, partially with my current position, why I've almost kind of made my own post to work between a bunch of institutions. And I still continue to work closely with the University of Oxford as well, really being at that kind of intersection of all the types of omics data to make the most of it. And I think if I hadn't had such a big data set for this paper, I don't know if I necessarily would have kind of come into that naturally.
Okay, Gabrielle, you have any thoughts?
Yeah, I think one of the takeaways I had from the paper is that when you kind of have your own research project, it feels very it's a project that you're focused on, and you kind of spend a lot of time you yourself thinking about it. But when you're actually writing and publishing a paper, you're often doing this with a lot of collaborators, and that just that process of working with other people is very useful in science, of course. Especially when you have like drafts of a paper, you send it to a collaborator and get very interesting inputs on how it might change. So you learn a lot through the process of writing, more so than you would if you just did a research project and publish it and then just moved on to something new. So I think the process of writing is useful.
Okay. Thank you very much, all of you. Thank you very much, the people that are there. It was super awesome to hear your comments. Your research is absolutely super interesting, and we're very happy to be here and to share this this webinar. So thank you very much. And it was a pleasure for me.
Thank you so much, Guillermo. And I'll close this down. As you've rightly stated, we've come to the conclusion of our webinar, Pathway to Publication and Impact. I want to take a moment to thank once again Hannah, Gabriel, and Allison for presenting today, as well as Guillermo, who did a wonderful job as our host. I'd also be remiss if I didn't send out a word of appreciation to IASP's Early Career Network, as well as Renee Chassie for their help coordinating this event. We'll be back here for the second webinar in this two-part webinar series on Tuesday, October 29th at 10 a.m. Eastern US time. So please do keep an eye out for more information as it becomes available. Thank you all for your participation today. Take care and all the best.